Okay, first I would like to thank Akoya for arranging this event. It's very good. We can learn the most recent advances and share experience using the technology. Okay, today's agenda. First, I'll give you an introduction about BostonGene, what we do, and then how we incorporate this fusion platform into our workflow, then our imaging analysis pipeline. And then later, I'll introduce some data, how we validate MxiF LDT for tumor immune profiling. Then a little bit on RNA sequencing correlation. Then it's a summary, then if you have any questions. So first, BostonGene. BostonGene started in 2015 as a bioinformatics company. But now we have a lab set up in Waltham, Massachusetts. We are a clear lab, CAP certified lab. So what we do is we do deep, analysis of tumor tissues. We have laboratory set up for DNA, RNA sequencing, for flow cytometry and imaging. So for all these, we can do a little bit listed here for RNA sequencing, DNA sequencing. And also, we can do liquid biopsy, including cfDNA, ctDNA, etc., etc. As you can even see here, we have TCR, BCR, repertoire checking. So we have a large team of bioinformaticians that will be able to do all this fancy analysis. As listed here, I'm not go over. I'll just say for every patient came in sample, we'll do this deep analysis and then integrate all this multi-modal data into solution treatment options, feedback to the physician to find the best treatment for the patient. So I'm responsible for the imaging part, so I'll focus on the imaging part. We use both tech platforms from Akoya in our lab, both the Opal, PhenoImager platform, and also this Fusion platform. So we adopted this technology because it can achieve high plex and with minimum tissue damage. I think it's crucial for this multiplex imaging. So from the early days, even from CODEX to Fusion, there have been many updates in terms of sample format and also the processing microfluidics changes. We are happy. Just say for the Fusion 2.0, I have a little bit of experience because we just introduced this one. It has dual flow cell. That means originally the time frame for one sample, we can now do two samples. And also we have a relatively flexible imaging format, which is important because we would like to have 16-bit images so as to have a larger dynamic range to analyze images. So this technology, the center, is to use DNA barcode conjugated antibodies, as I listed here. So all these are Akoya provided antibody ready to use conjugates. So we also conjugate our own conjugates, like the new ones listed on the right, and also depending on specific project needs. We also have to conjugate some of the older markers to a different barcodes and review in a different channel. For this, we have developed our own image analysis software and algorithm using artificial intelligence and machine learning. So here is a brief summary of the process. So starting from the raw images, we first do extensive QC checking because we understand the quality images will lead to quality data. So there's a QC process, and after that, we'll do segmentation. This is also use our own algorithm, and based on comparison, it's better than the, let's see, current available commercial ones. We are confident of that. For segmentation, then after segmentation, then the expression of various markers are identified and the cells are phenotyped. So for tumor and stromal region, we employ a mask approach. And also for some cells with an irregular shape, especially the fibroblast macrophages, these are headache for Euro, software approaches. For this, we employ a special method called tessellation and to deal with, and we can satisfy, get ideal numbers. So here to elaborate a little bit, on the left, these are all the images with different color denotes to single markers. We can see we can achieve a high plex, and using our software, we can phenotype cells into various phenotypes as listed on the right, and plotted in this UMAT plot. This is a very good supplement to the single cell RNA sequencing, which I know they have their own limitations. So next step, after all the cells are identified, then the next is to analyze the neighborhood. So that's which cell types are hanging together, which cell types are actually affecting either in a short distance, in a rather long distance. So based on clustering of these phenotypes, in the middle is an example of this tissue. As you can see, the yellow means tumor, and there are areas circled and magnified on the right, so you can see more detail. So we can see clearly they have distinctive cell composition. And for example, the first one is just tumor cells, and the second one, tumor with macrophages, T cells, and etc. This will have a tremendous different reaction, let's say, to immunotherapy or the tumor progression. So this is actually part of a poster we presented in last year's American Society of Clinical Oncology meeting. This, as the title states, okay, we have developed a clinical AI-driven MxiF imaging pipeline. This is an automated pipeline to characterize tumor microenvironment. I will not go in more detail, so if you are interested, you can later contact me. We can give you the full poster in which we have the whole process for validation of this method and its applications in several tumor types. So here I would like to talk about when we provide this tumor imaging, we will have this to be ready to provide to clinics. And here is what's planned to be included in that offering. So we have all this staining of the tumor tissue, and then we'll provide detailed information about the spatial distribution of immune cells and this microenvironment density, etc., etc. So for next slides, I'll just use one example to show that we can use immune cell penetration into the tumor as an index. So here is just the three examples. In the first one, we can see all the T cells, tumor cells, T cells are actually outside of tumor tissue. With blue is anti-inflammatory, red is pro-inflammatory. So in the middle is a different tissue that has infiltrated immune cells profusely. And the last one is immune desert, which you barely see any immune cells at all. We all know that in this case, the immunotherapy, the effect will be totally different. So this is a very valuable information. So all this information will be packed into a report for the physicians because everybody realizes they might not have extensive time to read hundreds of pages of information. So we have to do a high-level summary. As you can see here, it's only a few pages long. We have a main finding summary, and then followed by details like immune cell composition, even HE cooperation, HE incorporation. So the annotations from the pathologist, and then we have the overview of immunolabeling, and we have different detailed breakup of each markers. This is all this then fed to the physicians, so they know exactly what they're dealing with. So to push the assays, MxiF assays to the clinics, you kind of need to validate. So these are called laboratory developed tests. So per FDA definition, is a type of in vitro diagnostic test that is designed, manufactured, and used within a single laboratory. So this is us. So MxiF for LDT. We know the value of MxiF LDT because right now we know that for diagnostic and treatment purposes, a lot of single IHC has been employed in clinics. So if MxiF, which can stain multiple markers on one slice, why not? So it's the value is not questioned. There are different issues involved. For example, listed here, first, how to standardize different LDTs and how exactly to validate that, and how rigorous or robust the assay should be. And also, after you got these results, what's the standard form of reporting so as not to extend any study into a long paper. And of course, there are also regulatory hurdles, insurance policy, that's beyond me. So anyway, maybe a little bit ahead of time, we set out anyway to do our own MxiF LDT. This is also one way to test our MxiF assays. And also, we want to be the first one to get all this detailed information to the physician, to the clinics. So we designed panels for tumor immunoprofiling. And here, I just use one example. We actually have 7 panels targeting the most common 10 cancer types. So for this lung cancer panel, we include specific tumor markers like cytokeratin 7 and TTF1, thyroid transcription factor 1, and some stromal markers, and the rest immune markers, and some functional markers like Ki67 and PD-L1. So for this LDT validation, we constructed a TMA that consists of 20 tumor samples plus 2 control samples. So we use 3 spaced out slides for accuracy study and 3 consecutive slides for precision. For accuracy, we take into consideration our potential tissue heterogeneity along the depth of the tissue. So remember, for all the markers included, we'll have to do a single IHC for validation. So during this distance, there might be changes in the tissue structure. So that's why we have 3 slides included here. And for single IHC to compare with, we will compare that to the nearest MxiF slides, one of the 3. So for image analysis, we simply just use a free software, QPath, and whenever possible, we'll use percentage of positive cells. In the some stromal markers, which using this software, it's impossible to quantify percentage of cells. Then we use percentage of area. And then we use intra-class correlation, ICC, greater than 0.7 as a passing grade. So this is just a summary of a few markers. We have markers with good correlations as listed on left. Here we have a tumor marker, CK7 and TTF1. In the middle, stromal markers, vimentin and CD31. So the last one, immune markers, we just use CD8 and CD20 as an example to show. For accurate step, I just mentioned that there are 3 slides. So the one circles is the one used for comparison. They all correlate very well. And we naturally will have markers that's not correlated well. Here, for different reasons. So here are 2. One is a stromal marker. This is a fibronectin. We can see the correlation is ICC is only like a 0.4. The reason is that in MxiF, we see very confined immunostaining. But in IHC, you always see like a kind of a diffuse staining. Then it causes trouble because when you quantify, you have to use a certain standard, a threshold. For some samples, it's clean. For some samples, it's dirty. It's murky where to draw the line. That's why this discrepancies. Okay, next example is CD11C. CD11C has different issue because we noticed that in multiplex images, this antibody conjugate actually have this non-specific binding to smooth muscle actin. Although you can get rid of it using software, but that's not the right way. So we fail it. So as a summary of all the markers, as you can see, most of the markers passed the grade. According to current scheme, these several markers are not as good. So this provides a solid backing because we validated all these markers. Remember, for immune markers, we essentially validated across, let's see, 10 different tumors. So we have solid data to provide that demonstrate we have a solid assay for our offering. And right now, for fee-for-service, for big pharma, or for clinical trial, or various institutions. So outside this validation, we also used our own software to do detailed analysis. So shown here is one of the core that shows highly immune cell presence. So on the left is the cell typing. On the right is the cell community analysis. We can see we can do very detailed analysis. A different core showed a highly stromal region. So most of the page area are just tumor. On top left, you can see tumor region and with dense T self-infiltrate. So on the right, it shows. So for MxiF cell populations, we can use this UMAP to show. This is a dimension reduction plots in which you can see we can actually probe all these different cell types and do it in a systematic way. For this heat map, we can show that within the different distinctive communities, we have different enriched cell types. For example, the most obvious is tertiary lymphoid structure. We can see this B cell presence with some other cell types as summarized on the right. And for macrophages, we have CD68 and CD206 and other expression markers. For tumor, of course, you have this pan-CK here. Then what if we put all the slides, the results in one panel to see? This probably you will not have the chance to look at this. So all the slides, these communities put together, they give you a visualization of the potential variations we might encounter in real situation. you can see for every core, we have 6 slides used for accuracy and precision. Maybe you can see that every 6, they look similar. We can call it's from the same sample. But the detailed proportions, you can see there's variations, huge variations sometimes. So this variation could be from various sources. One is tissue heterogeneity across the depth of the tissue. Another, anyway, it's different experiment, different probing, different imaging. You are expected to see some changes. And also, the presence of some artifact might get your data a little bit off. So this is just to show, in the real situation, if you do validation, it could be quite different, quite difficult. So after the validation of this imaging experiment, we actually take out each individual core, extract the RNA, and perform the RNA sequencing. So this purpose of this is to find the correlation between data from these 2 probings. For one thing, everybody knows RNA sequencing in a high throughput analysis, you can analyze all these RNA transcriptome, but there's no spatial information. But on the other hand, if you use MxiF, remember all this data come from 1 slide or maybe a few slides. Can it be representative? We're not sure. So that's why we did this experiment. So first, on x-axis, they are MxiF communities. So the y-axis is RNAseq deconvolution method, which is based on the transcriptome profile to get an estimate of what cell types and what are the proportions in the real sample. We can see there are actually good correlations. For MxiF, immune cells here shows strong correlation with immune phenotypes from deconvolution, and other types too. It's interesting to see this T2, that means tumor tissues with a proliferating capability, the Ki67. So if it's a PD-L1 positive, actually you can see this community correlates clustered together with the immune phenotypes, but less with the tumor. So this is a very interesting part. The next part, we checked RNA sequencing. So essentially, we have different sets of genes that are always expressed together. We call it gene signature. So still, we compare these 2. I'm not going to other details, just to show that if you have PD-L1 positive, the proliferating tumor, you will be seeing all these immune active genes. And if you go a little bit further, you will see there are associated increased expression of these cytokine receptors as well as cytokines. So I'll just say it here, do a summary. First, the PhenoCycler-Fusion provide a powerful tool for multiplex immunofluorescence, enabling efficient, robust, and consistent marker detection on tumor slides. So we have MxiF LDT for tumor microenvironment immunoprofiling. And this validated and providing solid backing for our MxiF offering. And the last one is RNAseq deconvolution correlates with MxiF in reliable and consistent detection of signature RNA expression pattern and cell communities. Now, if you have any questions.
I'm here. Question from online. Awesome. I'm going to read it.
an anonymous attendee. It looks like BostonGene provides amazing information on tumor makeup and metabolism. You must use the best technologies available. What do you like about Akoya? Why do you use their technology over others? Wow, that is...
This is from Akoya.
I don't know. That's what I thought too. I don't think so.
Okay, as I mentioned earlier, we adopted this technology mainly because it can achieve a high plex. Just say other method can achieve this high plex too, but this one causes minimum tissue damage. That's what we like.
Awesome, thank you.
Yeah.
I have a couple more questions. Yue, who is from Akoya actually, she asks, if CD11C has non-specific binding with SMA, what other marker will you use for dendritic cells detection?
Currently, we still rely on CD11C, but different cell types, we essentially phenotype each cell based on not just a single marker, but a combination of multiple markers.
Awesome,
Yeah.
That's great. Catherine Zhu asks, how did you determine that your cell segmentation AI algorithm is high performance compared to the open source or commercial softwares? What was your training set?
I cannot go to very detail. I will just say this. We have manually annotated data sets that in exceeding of 20,000 cells from different cell types, and then use different software to do the segmentation, then compare the F1 correlation efficient, and actually our software achieve the best score. It's 8.84. Yeah.
That's pretty awesome.
Yeah.
Any other questions? How?
Yeah, this mic.
Had a question about your TMA design. So for each of the 20 samples, is it one core from individual tumors, or do you have multiple cores from the same tumor on that TMA?
No, one sample, one core.
Okay, great.
Yeah.
Thanks.
Thank you. Oh, hold on.
Yep.
Hi. You said you use one plex to compare, like with a MIF, like whatever MIF you get, you see one plex and compare the data. But what about if you have multiple markers, or if there is like a co-localization of two markers? So how do you do the analysis?
Excuse me, can you repeat that?
Yeah, there was a slide where you mentioned that when you have to see the markers,
Yes.
you do single plex and then you compare it with MIF, like multiplex, MxiF.
Yes.
So what happens when you have multiple markers, or there's a co-localization? There can be a discrepancy in the data, right? So that if you do the statistical analysis, or you go further in your study. So if it is a co-localization, so how do you detect how do you try to solve that problem when there is a co-localization, or if there are like multiple markers who are maybe 4 markers that co-localizing together?
I think the validation is based on the staining, not on cell types. So we don't care about if we co-localize. We just quantify that specific staining.
And my question is, when you're thinking about developing these LDTs using the high-plex panels, how do you think about the number of markers that are necessary to include in those? Or do you have like a standard set? Or, as the field's kind of starting to move into thinking of the utility
Yes.
of these higher plex, like how do you discern that between large data sets and necessary number of markers for your LDTs?
Yes, I think the size of the panel is depending how many markers can we handle at ease. Okay, all these markers being selected by our pathologist, and we could include more, but this is enough, and we want to get more markers. And commonly, I know in some studies they said even 6 or maybe 10 markers is more than enough. We at this stage we just want to get a more complex, more in detail analysis. That's why I went for that. Our panel average like 25 to 30 markers. We think that's the best we can handle.